Hello everyone! In the last video we imported our next generation sequence data, and in this video I will show you how we can find mitochondrial reads in this sequence data. A few videos back we downloaded some reference genomes of other stoneflies, and we're first going to start to import those into Geneious. Let's make a new folder and call this one reference genomes. We can import the data by drag and drop. Geneious will ask us if we want to have a sequence list, but we should keep them separate so we can work with each genome individually. I also included some single sequences from Mitochondria, which are from mitochondrial origin. Now we have to figure out which of those 4 mitochondrial genomes is the best reference genome to blast our hits against. We should pick one which has high similarity to our study organism. However, we don't have any sequences of Mitochondria, so we're just going to use the one we have available. This is another genus probably, but we're just going to use this one here as a blast query to figure out which mitochondrial genome of Plecoptera, which is available online, is the best hit, the best match, and which we can use to filter out some of the mitochondrial sequences in our dataset. To do this, we're going to copy the GenBank ID. Let's open a new tab, and we're going to blast. As always, you will find all the links in the YouTube description below. We select nucleotide blast. In here, we can paste in the session ID, and also in interest query, we should write down mitochondrial genome Plecoptera to narrow down our search. We want to use BLAST-N as an algorithm, as our sequences are probably not very similar. And let's open the results in a new window. Here are our results. Let's take a look into the table. And we see here that none of the sequences has a very good identity. However, the Plecoptera Muria has the highest identity, so for now we're just going to pick this one here and blast our hits against this reference genome. So let's go back into Geneious, and we're going to use Muria as a reference genome. To add this to a database, you need to go to first on Tools, Add Remove Databases, and then Add Sequence Database. I would recommend to use the same name as it is in the ncbi ID. So let me type this down really quick. And I also added Kami in the end to know which stonefly it is. We can click on OK. And now this was added to our database inside of Geneious. We can now go back to our sequencing reads, and let's just select the left ones. And you can go on sequence search, and in here we can select as a database our Kami database. Now we can also select the algorithm. The DC megablast is usually fine. You can also use the BLAST-N algorithm. Usually the BLAST-N algorithm will give you a few more hits, but it doesn't really matter too much. Then we want to export the hits into hits versus no hits, so it makes us 2 files, all the queries which were matched to the reference genome and all the queries which were not matched against the reference genome. This way we can use basically our reference genome as a template, map all the reads against it, and then we have our first dwarf genome for our organism. You want to make sure that the low complexity filter is turned off, because if you turn it on, Geneious will prompt you with a message that your sequences are too short sometimes, and it will not continue the search until you click on OK on that message. So it's a bit annoying. However, we can avoid this by not turning low complexity filter on, and then it will run the complete search without us having to click anything more. As an e-value, this is kind of an indication how good your hit is. We choose a minimum cutoff of -10. So very small hits, which are not very reliable, will be excluded and not transferred into the hit table. This all looks fine. We can click on OK. And now we see it starts the search. This will take a few minutes. It warns us here that some reads have ambiguities, but we can't really do anything about it, so we just click on OK. So here's starting the BLAST search, and I'm just going to fast forward and we see what we get. So we got our results back and we have around 2000 hits. Let's get our mitochondrial reference genome in here as well. You can just drag and drop it, and if you pressed Alt, you can copy the file. Now let's select the reference genome and the hits and go on to Map to Reference. In here, the reference genome is already selected. You shouldn't use the highest sensitivity because this will take quite a lot of time. However, iterations up to 5 times is recommended to optimize the mapping of the hits. Otherwise, you will get some mismatches and some gaps which are incorrect. Let's click on OK. We can see here that almost all hits were assembled. Let's take a closer look at the results. Let's zoom in a bit. And you want to make sure that highlighting is activated. If you take a closer look, even here some things are still a bit off, but we don't worry too much about these errors right now, because what we want to do now is we want to take a look at the CO1 gene. This thing here now, the consensus sequence, is the actual sequence of Mesocapnia. So the organism where we have our next generation reads from. We can now copy this one here. Just press Command C. And we are going to use this one now to search for the reference genome, because we have now information about the organism, not just the genus. And we can expect that by searching with this query here, which we just copied, we can find a bit better of a match to the actual reference genome. So instead of this accession ID, we paste in our sequences. We can keep all the settings the same. We also keep mitochondrial genome, and let's click again on BLAST. Here we have our results, and this actually does stay almost the same, but we see that also Pteronarchis is a very, very close match, so we could use both of them to assemble the mitochondrial genome. Now, as we have found our optimal reference genome, I think I'm going to use Pteronarchis to map the reads to, because it's kind of the most popular mitochondrial genome. We can now make one more trick. We're going to select all of those sequences here. They are all Mesocapnia related or reference genomes of Plecoptera, and we are going to add all of those into a sequence database for blasting. This might increase the chances again a little bit to find some additional mitochondrial reads in our dataset. Again, we go on Tool, Add Remove Database, and we're going to add this. And I'm going to call this Pleco mitochondrial genomes and Meso for Mesocapnia. Let's click on OK. Now this database was added, and we are going to use again the left reads and start a new BLAST search. In here we select our new database. The rest of the settings can stay the same, and we click on Search. This again will take a while, so I see you back in a minute. Alright, our search is done, and we see here that we get over 3000 hits. So we increased our hits by around 1000 by using several reference genomes. So basically all reference genomes which were available on ncbi for Plecoptera, as well as some sequences of the study organism. Now again, we're going to copy the reference genome. This time we're going to use Geneious. Just copy this one into the folder here. And again, we use map to reference. Let's click on OK. And here we have the first dwarf genome, which we can use for further BLAST searches. As we are going to use several BLAST searches and several mappings, I kind of want to make sure that I can trace which hits are from which BLAST search to get an idea what the progress is in each search, if we can add some new hits or if we can't. So what I would do is I would always annotate the hits from one BLAST search. So you click on the file, you click Command A, and now we have selected all of them, and we click on Add Annotation, and in here we name this one 0. So we just make it a running numbers, and we will take another motif each time to change the color. Of course, this should be undirected, and we can click on OK and on Save. Now let's assemble the hits again against the reference genome. We go on Map to Reference, and this should be again fine. We click on OK, and here we have our first mapping. Let's take a closer look. For now, let's disable the annotations we have just added, and we zoom in here. So, despite some mismatches, this looks really good. Also, we can now be sure that we have only one organism sequenced, because if it would have been several organisms of the same species which would have been pooled in the same sequencing one, you would expect some single nucleotide polymorphisms, SNPs. So, for example, one base here would have an A, and all the other ones would have a G. But we only get some rare mismatches. Those are very likely sequencing errors. So this should perfectly be fine. Let's take a quick look across the assembled genome to see if there are any problems with the mappings. You might get some gaps in here sometimes due to deletions or insertions, but this isn't really a concern. We don't see any strong systematic mismatches, so I think this is very nice. Maybe let's take a closer look at this one here. If you want to have an idea what is going on, you can also always disable highlighting to get a better picture. Yeah, it might be that we have some problem here with the assembly, but we can probably take care of this later on. Let's zoom out again. And I would say we can close this. What we are going to do now is we select the consensus sequence, and we're just going to export this consensus. So the consensus are basically all our mapped reads and kind of a summary. And we're now going to use this consensus and use this consensus as our first dwarf genome, and we add it to our database to map the remaining reads, the no hits here, against this consensus. We can expect that we find some more hits because the consensus resembles the real mitochondrial genome much closer, and most hits will have a 100% or close to 100% match to this dwarf genome. So let's right click and we click on extract regions. And here we have the consensus sequence. You notice we have also some gaps in here, but we don't mind that right now. And we're going to change the name to dwarfed1. Let's add this one to the sequence database. And we call this dwarfed1. Now we take our no hits, because here there could be still some more mitochondrial reads in here. And we're going to go on sequence search as before, select our new dwarfed genome, and we again click on search. So I see you when the search is done. Okay, our blast search is done and it has found 600 additional reads. Let's select again all of those and we're going to add an annotation. And this time we call this one 1. Let's select something else in here to get another color. And again we have this undirected. Click on save. And also let's maybe rename this one v1. Also we can make a new folder in here. And let's just store all our relevant data in this folder so it doesn't get too messy. We copy our hits. We copy our hits v1. And we are also going to copy the reference genome into this folder. Let's maybe call this one version 0. Alright. Let's now select all 3 of them. And we again use map to reference. Let's take a closer look at the assembly. And I'm also going to get to annotations and activate this one here as well. Let's zoom in a bit. And we can see right away that in the second iteration of our blast search, we could add some more reads. And if you take a look at the colors right here, you see that the first one here was the dwarfed genome. And in the second blast one, it was able to extend the edges where we had no sequence information by a bit. This gets really complicated because our sequences are only 100 base pairs long, so it takes probably a lot of iterations, and also the blast algorithm has a harder time assembling short reads than with, for example, 454 data, where reads are often longer than 400 base pairs. Let's again uncheck the annotations here and zoom in to this one to check if this is assembled correctly. Let's turn on highlighting. And you can see here that this maybe is not correct. So it's probably better to delete those sequences here from the assembly because they are not matching very well. If you would keep them, we would have wrong sequences in our dwarf genome, and BLAST would find even more wrong sequences which are not of mitochondrial origin. So it's important to check your dwarf genome every assembly step. If you want to delete sequences, just select them and we click on delete. Also, this one doesn't look right. So you see this now looks a bit better. Okay, let's now check the complete genome if we find any other errors. This here also doesn't look correct. Looks like we have a microsatellite in here, and this is a hit which is quite common because the microsatellite, this repetitive pattern of TNA, is quite common in the genome, so we should definitely get rid of all those sequences. Again, we're going to delete them. Let's turn highlighting on again. And the rest looks okay. Not sure about this sequence as well. Let's delete it for now. This looks good so far. Maybe this here is also a problem. Yeah, it's difficult to decide which sequence here is right or wrong, because this one here is matching quite well. So we're just going to keep it for now. Those sequences here also don't look too good. We delete them as well. Okay, now we save this document. And also let's rename this one maybe to hits version 1, edited, so we know it's associated with this last one. If we do now further assemblies, we should add additional reads to this mapping right here, which we now have edited. If we would choose all the hits again, we would probably get the same errors like with the microsatellites again. Let's again make the consensus sequence. You can do this under Tools, Generate Consensus Sequence. Let's click on OK. And we call this one Draft 2. Again, we're going to add this to the database. And we're going to go to our no hits and run a new blast search against Dwarfed version 2. Here are our hits, and I have already annotated them. What worries me a bit is that we only found 48 sequences, but let's assemble them to the dwarfed assembly and see where they are clustering. We move this one again to our Mito assembly folder and rename this to hits version 2. Let's select hits version 2 and our assembly, and we go on map to reference. Make sure that you don't activate dissolve contigs and reassemble. We keep it as it is. As we don't reassemble, we also have no fine-tuning. We can click on OK. If we compare both numbers here, we see that 23 hits were assembled. Let's rename this assembly to edited as well. Let's take a closer look. We have some hits which are assembled here and here. So this doesn't matter much, but this is very nice, as long as the hits are looking okay. Let's also disable annotations, but those two hits here, they were added and they look really good. So that's nice. Also here in an area with low coverage, additional hits were added. I just realized that the annotation I used to get the 2 on those hits doesn't let them highlight. So let's zoom in here and see if they are looking good. Doesn't look exactly right. Actually, let me do this again and I add an annotation which allows highlighting. Give me a second. Okay, here we are again. And we can see that this annotation here are the new hits. However, this doesn't look really good, so I think we should delete these sequences again. This is a lot of A's, and this doesn't make sense because this region here is gene coding. So we're going to delete these sequences here. I'm also going to try and delete maybe this sequence here, as it might be preventing other maybe correct hits from being mapped. Let's click on save again. And let's see if we find any more additional hits. it did find some matches over here, but those also don't look very reliable. We delete them as well. Alright, except for one spot, which was this one here, we didn't get a lot of new hits, which is a bit unfortunate. Could have two reasons. BLAST might be not able to extend those regions here because the overhang is not big enough, or actually it might be a region where we have low coverage. So to test this, we used all the time the left set of sequences, and now let's try and use the right sequences, so the 500,000 sequences from the right side of the paired end reads, and we're going to blast those against our initial reference database, which included all the different mitochondrial genomes of Plecoptera which are available. And then we're going to use the hits we have from here, from the left side, and the hopefully new right one hits, and add all of those together and assemble those again against our initial Geneious reference genome. This way we can check if those values here are due to low coverage, because then they would be filled, or due to problems with the dissimilarities of the sequences. So let's do this now. We're going to click on this one here, on the right one, and we go on sequence search, and we select Pleco mitochondrial genomes. The settings can stay the same. The BLAST search is done, and we have here our hits, around 3000 sequences, and we're going to rename this document to hits right. Let's also move it into our Mito assembly folder. Let's also add some annotations onto those sequences here. We again press Command A, add annotation, and we name this one R for right side. You can click on Save. And now we select all hit files and our reference genome from Geneious, and we're going to map to reference. Make sure to iterate up to 5 times, and you can click on OK. This now will take some time, so I'll see you back in a second. Here are our results. Let's take a closer look. We also activate the source annotation. Alright, source is already occupied in our reference genome as an annotation, so we're just going to delete this annotation here. And now we can see the actual coverage. The line in purple and the line in blue are the colors of the left and right BLAST ones. And as you can see, they both have roughly the same pattern. So by adding the right reads to the dataset, we didn't gain any more information. This indicates that it's very difficult for the BLAST algorithm to map those reads on the edges. So we probably have to try something else. So let's save this for now. Let's annotate regions which have good coverage and are very likely correct with an own annotation in the consensus. Actually, let's just hide the annotations we made, and we keep the annotations of the reference genome, so we have an easier time adding our annotations. This should be fine. Let's zoom in a bit more. So to add an annotation on the consensus, we select the region we want to add the annotation to and click on Add Annotation. Let's select an annotation which isn't used already, and we want to have this undirected. Let's call this OK. Now we can extend the annotation how we want to have it. This is all looking pretty good. But here we have regions where we are not too sure anymore. From the first look, this looks quite similar, but it probably isn't, because the overlap here is very short. So we're also going to delete those sequences here. And we probably can rely on those down here, because they have a good overlap with the rest of the sequences. Also, if we take out the read quality, they are looking fairly okay. So up to here, the sequence should be okay. Let's save this one. Then we have a big gap. Those sequences here don't make too much sense. But they are here probably correct. So let's delete this one here. And we move those one backwards. Well, actually we can keep them because the consensus has a gap here as well. So this will be removed automatically. So we can go ahead and add a new annotation. I will finish this now for the complete genome and show you any interesting spots, if there are any. The sequences for ATP8 could be interesting, because if we take a closer look in here, we see, if we disable highlighting, that we have quite a lot of T's in our sequences. So they match quite well here, but this kind of looks suspicious. So as this is coding, we can just run a translation. And you can see here, let's turn on highlighting again, that this isn't very closely matching. So those sequences down here might not be very reliable. However, if we just take a look at the color itself, the amino acids are kind of similar which are used. So it's a difficult decision if we should exclude it or not. I would say we add an annotation which is saying unsecure. I found this other spot where it has a lot of A's in the sequence, so this is also not really possible. So we're going to delete those sequences as well. Overall, the first view of the data doesn't look too good, but we will see what we can make out of it. So let's delete those sequences here as well. Okay, I'm done adding the annotations, and we're going to select the consensus sequence and save this one as an extra file. Let's minimize everything again, and we should rename those two here to HITS version 3. I think it's a good idea to take some other reference genomes and make an alignment with our dwarfed sequence to see if the at least protein encoding regions are making sort of sense, or if there are more errors in our sequence. We especially have the problem with areas of low coverage and some mistakes in the mapping, where the blast search has detected some reads where it was thinking it's mitochondrial, but it probably isn't, so it doesn't fit very well. So at the moment I'm not exactly sure if our method is suitable for actually assembling a mitochondrial genome when the reads are very short, but we are just going to continue working on it and see if we can get something useful. If it doesn't work, we probably have to search for some alternative methods. So let's get some more reference genomes in here. And actually let's include also the Mesocapnia sequences. Let's maybe make a new folder. And we call it alignment. just to have things a bit more organized. This should be fine. Let's select all reads. And we're going to go on multiple align. We can try to let it detect the sequence direction automatically. Let's see if that works. Otherwise, we have to form the reverse complement. Let's take a look at the alignment. We see here 2 sequences didn't really work out, so we can delete those. And then let's zoom in here. Let's also add some annotations. And we see here the whole thing is reversed. So we click once on reverse complement to have it in the other direction. Let's include the translation and let's save this for now. And our reference genome was the NC one. So move it down here. Let's also add an annotation in here to say that this is a coding region. We select CDS. We see that we have here some gaps, which is not supposed to happen. Let's allow editing and remove the gaps. And if we look at the amino acid pattern, it looks like we are missing some bases. Yeah, it's really difficult to say what is going on here. Alright, I would say we call it a day for now, and I will try to figure out what is going on, what is the problem with the assembly, and we discuss the results in the next tutorial. At the moment, this isn't working exactly out as planned, but at least we should be able to figure out what the problem is, and maybe we find a solution, even if the solution is using another program or a proper assembler. So, see you in the next video. Bye bye.